Now, the whole rest of this unit and this note packet is about osmosis, the movement of water, and how the movement of water can affect cells. And the movement of water is so important, and it has such an effect on our cells that we dedicate this whole last part of this unit to the movement of water, to osmosis. Now, I'm going to go through some rules to help you predict which way is water going to move. And you already learned the first rule is that water always moves from an area of high concentration of water to an area where water is in low concentration. And you've seen this diagram before. There's a high concentration of water here. There's a low concentration of water here. So there's going to be a net movement of water in this direction. Now, sometimes I just show you water molecules like this, and you can see that there's definitely more on this side than on this side. But sometimes we just give you water concentrations, and we say this side, or solution A, is 90% water. Solution B is 50% water. And you have to just remember this rule that water moves from high concentrations to low concentrations of water. So in this situation, water is going to have a net flow of water in this direction. Now, the reason why I have to teach you other rules is because sometimes we do not give you the percent of water. If we don't give you the percent of water, you're going to have to apply some other rules. So here's rule number 2. Sometimes we say that a cell or a solution is hypertonic or hypotonic. So then you have to have this next rule memorized. You have to remember that water is always going to move from a hypotonic solution to a hypertonic solution. Now, to help you memorize that, and trust me, someday when you're sitting in your college biology class and they're talking about hypertonic and hypotonic solutions, you're going to remember this rule. So imagine if you were going to go to a party this weekend and you have 2 choices between which party you want to go to. One party, you text people and they say it's kind of boring. You text someone else at a different party and they say it's a great time, everyone's having fun, everyone is hyper. You're going to go to the hyper party. So remember that water is going to go to the hypertonic solution. So let's just write over here to help you remember this rule. You want to go to the hyper party. Now, there's really no such term or no such thing as a hyper party when we talk about biology and in science. So don't write that water goes to the hyper party. You need to remember water goes to the hypertonic solution. We're just going to use this hyper party to help you remember this rule. Now, when I'm talking about hypertonic and hypotonic solutions, then I'm talking about how much water there is compared to solutes. So let's review real quick. If we have dissolved molecules in a solution, they're called the solutes. And remember, water is sometimes called that universal solvent because it dissolves so many different molecules. It's called the solvent. Now, a hypertonic solution is a solution that has a higher concentration of solutes compared to another. So it has more solutes dissolved in it compared to another. So imagine if you have tea and then you have a glass of tea that has sugar in it. The one with the sugar in it is going to have more solutes in it. It would be the one that is hypertonic. Now, to help you remember that, I like to think of that old wives' tale, that if you give kids a bunch of sugars, then they're going to be more hyper. So think of more solutes, like sugar. That's going to be the hyper one. So by default, the solution that has a lower concentration of solutes is going to be the hypotonic solution. And anytime we have 2 solutions that have the same concentration of solutes, then we say those 2 solutions are isotonic. You probably know what that prefix means because in geometry you probably learned what an isosceles triangle is. And that was a triangle that has 2 equal sides. So iso when we see that prefix iso, we're talking about something that's going to be equal, or two things that are going to be equal. So these are examples of problems that we can ask you, and we're going to ask you to predict which way water is going to move. So we made a solution, and that solution was hypotonic, and we placed it in a dialysis bag. And a dialysis bag is kind of like plastic Ziploc bag, and it's got small little holes in it. So if the inside is hypo, then the solution that we place on the outside by default is hyper. Now you have to remember, water is going to go to the hypertonic solution, just like you're going to go to the party where everyone's hyper. So we're going to show water's moving out into that hypertonic solution. So let's label that arrow with H2O so that we know we're talking about water moving, because we're talking about osmosis. So if I give you a situation, I say a cell, so here's a cell, is surrounded by a hypotonic solution. Predict the movement of water. In this situation, water is going to move into the cell because the inside is that hypertonic solution. Water always goes to the hypertonic solution. Now, just because a solution is labeled hypertonic, it doesn't mean that it's always going to be hypertonic. So, for example, if I have just plain water compared to a 3% sugar solution, obviously water is going to be the hypotonic solution, and sugar is going to be, in comparison, the hypertonic solution. Now, if I take that exact same 3% sugar solution, if I compare it to a 10% sugar solution, then the 3% sugar solution now has less solutes in it. It is the hypotonic solution, because that 10% sugar is the hypertonic solution. So when we're talking about hypertonic and hypotonic, those are terms that we use to compare different substances. So just because you label a solution hypertonic doesn't mean it's always going to be hypertonic. It's just like you might say I'm short, but if you compare me against someone that I'm taller than, then I'm no longer short, I'm tall. So that was rule number 2, and now we're on to rule number 3. So take a look at this diagram that I've given you right here, and you can see that I do not give you water percents. I'm not showing you that's like there's 90% water or 50% water, and I'm not showing you there's a hypertonic and a hypotonic solution. I'm giving you solute amounts. If I give you a problem and I ask you to predict which way water is going to move, and I just give you solutes, you have to remember this rule that solutes suck water. Wherever there's more solutes, then that's the way that water is going to move. Water is going to move to where there's more solutes. So think solutes suck water. So here's a U-tube, and I have a membrane that's separating solution A and solution B. If you add up the amount of solutes over here, it's 0.9 molar. Over here, we have 1.2 moles of solute. So there's more solutes on this side. Solutes suck water. Water is going to have a net direction or net flow from side A to side B. Now, some of you maybe picked up, well, I already know that if there's more solutes, this side is the hyper one, and this one's the hypo. And if you thought about that, that's great. So if you know that this side is hypertonic because there's more solutes, water is going to go to that hypertonic solution. So look at some other diagrams here. So it looks like we have all water over here, and we see we have some solutes over here. So this is an easy one. Since there's more solutes over here, solutes suck water. Water is going to move in that direction. You also maybe determine this side is hypertonic compared to this side. And water goes to the hypertonic solution. A few more. So let's predict which way, again, water is going to move. There's more solutes inside. Solutes suck water. So let's show water's moving into that cell. And we're going to do some math here. We have 45 grams of solutes inside, 30 solutes outside. So there's more solutes inside. Show water moving in. That inside is hypertonic compared to the outside. Now on this bottom diagram down here, you can tell that it looks like the concentration of solutes is the same both inside and out. So if some water moves in, some water is going to move out at the same rate because these two solutions are isotonic. Remember, solutions at the same concentration are considered isotonic solutions. And then one more diagram. It says we have pure water on the outside. That means it's 0% solutes. Obviously, we have solutes inside. This would be hypertonic. This would be hypotonic. Solutes suck water. Water is going to move into that cell.